Yeah, good morning to all of you. Can you all hear me properly? Rosila, can you hear me? Right, we have some little amount of MCQs remaining in the Ananda College paper also. So what's the last question that I discussed in Ananda College paper? Question number? Last question? 29. Okay. Right, Rosila, can you hear me, Putte? Right, so we'll quickly finish the given MCQs. දෙරණ කතා කරද? ඔව්. ආහාර දැන් හරිනේ. හරි. So we'll be discussing the remaining MCQs here. 29th MCQ, answer number 2 is correct. Nucleosome contains 146 base pairs. That is the incorrect statement over here. Select the correct combination. Answer number 5, that means E. All new cells arise from pre-existing cells. Rudolf Virchow gave that idea. And question number 31, incorrect statement regarding polyploidy. Polyploidy organisms are always non-fertile. So answer number 1 is incorrect, because you know, if the polyploids are having, let's say, some even numbers of ploidity, 4 and 6 and like, they are fertile. And especially in plants, polyploids are possible. Animals, polyploids are usually not possible. So these are fertile organisms. Right, next one. 32, select the correct statement. DNA can answer number 5 is correct here. Yeast artificial chromosomes, yeast cloning vectors carry autonomously repeating sequences of centromeres and ori. So answer 5 is correct here. Answer 13. If you have any issues, please ask. Okay, I'm just going through quickly because we want to go to the second paper also today. Correct statement regarding mutation is answer number 5. A segment from one chromosome break and then change its orientation and can be fused to the same chromosome. Okay, that can happen. After this one, I'll do little revision about mutation types also. Hi, good morning Onit and good morning Haravindi. You can hear me, right? I'm just discussing the last bit of Ananda College paper. If anyone don't have the paper, just let me know. I'll forward it. Send me a direct message so that I can send it to you if you don't have the paper with you. I'm now in question number 34. In an ecosystem, Answer is answer number 3. Minerals flow in cyclic manner. In an ecosystem, energy flows in unidirectionally and minerals are recycled and used again. So answer 3 is correct there. Which of the following statements regarding tropical forests is incorrect? There are two types of tropical forest, tropical wet forest and tropical dry forest are there. Tropical rainforest are the tropical wet forest. Tropical rainforest and tropical dry forest are there. So answer is answer number 3. It's not evergreen. Please change it into evergreen. The word should be third sentence. Evergreen plants are present. That's wrong. Tropical dry forest, deciduous plants also present, not only evergreen. Evergreen means throughout the year you have green color in the plants, and the deciduous means sometimes of the year the leaves will fall. Okay. Next one. Which of the following is not a characteristic of pathogenic microorganisms? Answer number 4. Ability to enter directly into host blood by disrupting host cell. It's asking not the characteristic of pathogenic microorganisms. Other answers are correct there, no? 37. Which of the following response correctly indicates antibiotics and their functions? Answer 3. Tetracycline inhibit the protein synthesis. That's the correct answer here. 39. Question number 39. Answer is 2. Regarding correct statement regarding dengue vector. Adult female mosquitoes have white markings on its body. One minute. Next question. Question number 38. Which of the following statements is correct regarding solid waste management? Answer 3. An anaerobic condition in the pile of compost promote the activity of methanogenic bacteria. Correct? Next one. Select the correct statement regarding dengue vector. Answer 2 is the correct one here. I discussed that one. 4th here. Not an important of tissue culture, importance of tissue culture, it should be. Not an importance of tissue culture. Answer number 3. Ability to produce plants with variation. All others are correct here. But even if it is not mentioned in your syllabus, tissue culture is sometimes used to get variations also. You can maintain a culture of cells and you can induce variations by adding chemicals such as colchicine. You will be inducing variations in the tissue culture technique also. But tissue culture technique is well famous for in vitro multiplication of plants to get more plants which are equal to each other. during a short period of time without getting influence of the external environmental conditions. Hi, next one. 41 to 41 to 50, I'll read the answers, just mark the correct answers there. 41, answer 5. The correct answers here are A and C. 42, again answer 5. Correct answers are A and E. 43, answer number 2. 44, answer number 1. 45, answer number 5. Only C is correct. 46, answer 3. 47, answer 1. 48, answer 2. 49, answer 5. A, B, C are correct. 50th, answer number 1. Right? I'll write the answers again, 41 to 50 again. Please go through and ask if you have any issues. If I want to discuss any of the questions, you can let me know. Rilki, Losilu and Omit, anyone? If you have any questions, please let me know. Wait, 47. Select the incorrect statements. Inbreeding is used to produce pure breeding generation. That means self-fertilization is done for several generations in order to get a uniform progeny. That is true. Inbreeding is giving uniform pure breeding generation. That is true. Outbreeding allows desirable traits of the exotic parent, which the indigenous parent does not have, to be transmitted to the progeny. That means if you have certain character in one particular plant, you will be crossing with another plant or one of the parent of these plants in order to introduce that particular character into the next generation or the new from the exotic parent to the indigenous parent. So that is also true, Pute, okay? So outcrossing is giving the characteristic to the other one also. C, the progeny obtained from interspecific breeding is partially fertile. It can be partially fertile. It may be fertile also, infertile also it can be. So that I took it as wrong, okay? Not always, no. Outcrossing performed between two species. That is true. Inducing advantageous mutations in plants occurs in mutation breeding. Ekath harida. incorrect ahane neda eke 47 1 da man gatte 47 5 it should be neda puthe incorrect 1 ahanne ne incorrect ekai neda enne last ekath correct 5 ne yeah what did you get puthe dilki 5 da 47 47 5 it should be. Hari, any other questions? Fiftieth. Which of the following statements regarding rhizosphere is correct? Symbiotic interactions on between plant roots and microorganisms is correct. Bacteria are the most numerous organisms in the rhizosphere. True. Pathogenic microorganisms are absent in the rhizosphere is wrong. It should be rhizosphere, not cystosphere. It's wrong. Pathogenic ones are also, they are good ones and bad ones. Both are there in the rhizosphere. Symbiotic fungi associated with the rhizosphere, true. Microorganisms in the rhizosphere not feed on the root, wrong. They are feeding on the root exudates, so that it is wrong. So answer is answer number 1. Okay. Today we are going to make a summary of mutations also. I'll be quickly doing a little summary about mutation part also so that you will memorize again. So mutation is an alternation. They should be permanent alterations, okay? Right? So alternation of nucleotide sequence of the genome of an organism. This should be a permanent alteration. We should go to the next generations as well. So I'll be dividing the mutations into 2 main categories. Don't write, wait a little. What are the 2 main categories of mutations, Putal? Gene mutations and chromosomal mutations. Gene mutations are small scale mutations. It may be either one pair of nucleotides or else few nucleotide pairs. Right? So if it is one pair of nucleotides change, what is the name given? They are called point mutations. Right? Point mutations if only one nucleotide pair is altered. So these two commonly we are having few types of nucleotide pair changes. So number one is substitution. What is the meaning of substitution? If I draw a nucleotide sequence like this, A, T, A, C, A, G, C. C, A, T, G, T, C, G. Let's think. A and T is removed. It will be substituted by another pair of nucleotides. Let's say it is substituted by now guanine and cytosine. So this is the substitution. One pair is removed and it's substituted by another nucleotide pair. Understood now? Right. Substitution is one thing. Number two, insertion. What is insertion? The previous version is there, but some more is included. That means there was A and T here, and you'll be introducing another nucleotide pair here, like this, right? Insertion. The number of nucleotides increase now because of insertion of one pair of nucleotide. It may be more also, right? Both can be present. Number three, deletion. What's happening in a deletion? One pair is completely deleted. Okay? Or one pair of few nucleotides. Okay? Understood that part? That part okay? Right. So we have few examples of chromosomal, gene mutations, diseases or disorders coming up with the chromosomal mutations. What are the two examples given in our book? Number one, color blindness, red-green color blindness. Number two, sickle cell anemia. Okay. Right. Now everyone quickly go to your books. Do you have the book with you? Read gene mutation part completely and finish. Everyone come and read. Page number 24. Okay, Pute? Right. Then I'm going to divide this chromosomal mutations into two types. Alternations in chromosomal structure. Number two, alterations in chromosomal number. Correct? Alterations of chromosomal structure, there are few types. Look at this one now. Right. Number one, deletion. Deletion, what happens here is part of the chromosome is completely deleted. Let's say this is the chromosome. One part is completely deleted and this part is rejoined, right? So that is called deletion. So one set of genes can be lost because of that. These are big scale mutations than that side, okay? Right. So number two, translocation. What happens there is, yeah, segment of one chromosome is transferred to the another. Part of this is broken and it's now joined to another chromosome so that there is some repetition of, there is some more number of chromosomes in one chromosome, the other one having less number of genes. Some genes are lost from the chromosome, the other one is having more genes. And number three, duplication. In duplication, part of the homologous chromosome is broken and it's given to its the homologous other partner, and so that like homologous chromosomes are having same type of genes, no, the recessive and dominant alleles are there, no, so these will be same genes, so that it will be some repetition of genes when it comes to homologous partner. That means one part of this chromosome is going and joined to this one like in the previous case, but it's the homologous partner here, right? Then එතකොට repeat වෙනවා from genes, right? For example මෙතන A let's say මේ level එකකින් capital A capital B තිබ්බා, මෙතන simple A simple B තිබ්බා කියලා. So now මේ part එකක් break වෙලා යනකොට මෙතන capital A B අලුතෙන් ඇවිල්ලා. කියන repetition එකක් හරි. Number 4, inversion. Part of the chromosome is broken and turn upside down and joining again. So that is called inversion. Here now read that part also, alterations in chromosomal structure part. Read completely. Alternations in chromosomal number. There are two types. It may be aneuploidy. Aneuploidy means one chromosome is more or one chromosome less. And polyploidy means number of sets are increased. Alright, so aneuploidy again we can divide into two types. Monosomy. Monosomy means usual number 2n minus 1. ඒ කියන්නේ අපේ 46 minus 1 කියන එක තමයි monosomy කියන්නේ. එකක් අඩුයි කියන එක. Alright. Trisomy වල වෙන්නේ මොකද වෙන්නේ? 2n plus 1 ඒ කියන්නේ 46 plus 1 කියන එක. x y ඔය වගේ. මෙතන තියෙන x not ඒ වගේ. Right? Polyploidy වල වෙන්නේ more than 2n. That means 3n, 4n ඔය වගේ 6n repetitions තියෙනවා. set එක පිටින්ම වැඩි වෙලා. right? තේරෙනවද වැඩේ? හරි. ඒ side එකත් කියවන්න and now you can copy the chart also. Right, now you all can copy the chart quickly. Finish writing? All done? What about others, Puthe? All done? Okay. Onit is asking what is ploidity level? That means we are diploid organisms. If we take our one of the gametes, egg cells or sperm cells, those are haploid. So usual numbers are haploid and diploid, but there may be unusual things, the triploid, tetraploid, hexaploid. Likewise, ploidity number can be increased. That means they're having more than two sets of chromosomes. It's kind of abnormal. It's a mutation, right? Usually, this is not much common in animals. It's common in those plants mostly. It's not remaining constant in aneuploidy. It's kind of a big mistake if it is aneuploidy. You know, for example, Turner syndrome is because of monosomy. Klinefelter syndrome is because of trisomy. So abnormal children are born because of this monosomy and trisomy. Even the Down syndrome, it's a monosomy in one of the autosomal chromosome, not sex chromosome, autosomal chromosome, right? Hari? And there's little thing about gene mutations. We have to discuss about those silent mutations and all that. I'll discuss that part also. Right. Let's say particular nucleotide sequence is there. A, G, C, C, A, C, G, C, A, T. Let's say so each of these are coding for particular amino acids. So let's say there is a particular sequence of amino acids like amino acid 1, amino acid 2, amino acid 3, amino acid 4. Let's say because of certain mutation, this adenine is replaced by particular other nucleotide, let's say guanine, right? So one chance is, you know, like some amino acids are coded by more than one codon, so that there may be a chance that it's not altering the given amino acid. meaning that there is a mutation has happened, but there's no change in the polypeptide sequence. That kind of mutations we call it as silent mutations. That means there's no alternation of the sequence of amino acids, so that there's no change in folding. Correct folding will happen and correct protein will be given out, so that no issue even if there is a alteration in the nucleotide sequence, right? But there may be some incident, let's say this is filled with another amino acid, but another nucleotide, this is changed into another amino acid here. So that there is a slight change in the amino acid sequence now, right? So that this will affect the correct folding of this particular amino acid sequence, so that the function of the protein will be affected, right? It may be affected. For example, in the sickle cell anemia, one amino acid is replaced with another because of one single mutation, right? So that it's changing the sickle cell, changing the usual shape of the red blood cell into a sickle shape, which is kind of some distortion has happened in the protein. So this kind of mutation we call it as missense mutations. Missense, right? Wrong sense. And then there may be a chance addition of some nucleotide introducing a stop codon. If it is introducing a stop codon, premature termination of the amino acid sequence would happen. That means it's not lengthy as usual. It may be stopped at the point where there is the mutation. So that kind of mutation we call it as nonsense mutations. Got it? And another type is, we call it as frame shift mutation. That means reading frame would be changed because of some substitution like mutation, right? So that this is the correct way that we read earlier. Let's say another one is added here, so that you'll be reading from here now. From here onwards, the complete amino acid sequence would be changed, so that the protein is completely altered. Can you understand? I didn't get you on it. In the book, it says fluidity level has not changed. No, Pute, this type of mutations, only the fluidity level is changed, not in trisomy and monosomy, fluidity level is changed. Okay? I didn't tell that. Polyploidy is another type of mutation, not the aneuploidy. Okay? Aneuploidy is a different type. Okay? I now read about that also completely. The silent mutations, nonsense mutations, missense mutations, frame shift mutations. First paragraph in fourth page number. Yeah. If you put the fluidity level is changing, this type of mutation only. Polyploidy is one type, aneuploidy is another type. In aneuploidy, nothing is changed. Okay, fluidity level is same. Okay, it's 2 in people. You have misunderstood, I guess. I'm erasing this side. Hope everyone have done. I'm writing the question to write after the class and submit to me. Everyone should submit it to me after the class. Question number 1, part A. What are mutations? Part B, you all have to write it in a paper and submit. Part B, what are the major causes of mutations? Part C, explain giving suitable examples the different types of mutations occurring in living organisms. Write down the question and keep. You will be writing after the class. Done writing? Let's go to the given paper to you guys. I have given a paper from Pre-Med Association. Paper 2 will be discussed today. First question is asking, adaptation is a characteristic feature of life found in living organisms. What is the definition of adaptation? You have to write these things. There are 3 points given here. Adaptation is a peculiarity of structure, physiology, or behavior that promotes the likelihood of an organism's survival and reproduction in a particular environment. Again, I'm reading. Okay, I'll write it here. Peculiarity of structure, physiology, or behavior that promotes likelihood of organisms survival and reproduction in a particular environment. Right? Give 2 examples for adaptation. Number 1, sunken stomata in mangroves, xerophytes. Xerophytes are the plants which are living in less water conditions, and viviparity in mangroves. What is cell cycle controlling checkpoint? Give 3 phases in the cell cycle which cell cycle controlling checkpoints are present. Checkpoints to ensure that the cell is ready for the moving to upcoming phases of cell division. Correct? Did you write that? Mark if it is correct. And G1, G2 and M phases are having checkpoints. Right? I'll tell you how marks are distributed here also. Structure and physiology, 1 mark. Here behavior that promotes the likelihood, 1 mark. Reproduction in a particular environment, 1 mark. Okay, so 3 points are there in that. Here, with sunken stomata, viviparity, 1 mark. And also you can write about splayed out footing camels also here. And here, checkpoints, that definition 1 mark. And this one 1 mark, okay. Write 2 differences between the division of cytoplasm in plant cells and animal cells. Plant cells cleavage furrow is formed. Sorry, animal cells cleavage furrow is formed. In plant cells, cell plate is formed. Next one, give 2 reasons for the importance of photoprotection. Photoprotection is given by which pigments, Uday? Which pigments are giving photoprotection? Carotenoid pigments are giving photoprotection. That means it's preventing the chlorophylls from bleaching. So it says the importance of photoprotection. Number 1, protects chlorophylls from excessive light energy. Number 2, prevents the production of reactive oxidative molecules which are dangerous to cells. Right? Next one is asking what is aerobic respiration. So, process of synthesizing ATP from the respiratory substrates such as glucose in the presence of molecular oxygen. I'm writing, check whether the given exact definition is there in your answers. The process of synthesizing ATP from the respiratory substrates such as glucose in the presence of molecular oxygen. State the location, net yield of ATP and the amount of NADH produced during the process of glycolysis. Location is cytosol. ATP net yield is 2. NADH produced 2 NADH. Are 2 NADH. Mark if it is correct, 1 mark each. A student is planning to determine the rate of aerobic respiration in the in the laboratory using germinating green gram seeds. Draw the apparatus that can be used for this purpose. So, what is the apparatus used here? Respirometer. So I'm going to draw and show you the respirometer here. And I explain that also quickly. We'll be putting germinating green gram seeds over here. There will be an ignition tube with potassium hydroxide and there's a tap. This is connected to a U-tube. There's a colored liquid here. So you have to label everything. Just draw the apparatus, then I'll explain how to use this apparatus. Done. Finished drawing? Right. So what we do first here in this experiment, you have to soak the mung beans in water and then you'll be keeping it to germinate. And after that, you'll be removing the green color parts of the seeds and then you'll be placing a non-weight like 25 grams of mung seeds here. And then you'll be inserting a ignition tube with potassium hydroxide and then you'll be closing it and you'll be sealing it with Vaseline. The apparatus is sealed with Vaseline. And then initially you'll be bringing the water levels to one level. So you'll be marking the initial water level. And then you'll be keeping it for like 2 hours to respire. And then you'll be measuring the changes in the water column. And this is taking oxygen for the respiration and giving out carbon dioxide. The given out carbon dioxide is absorbed by this KOH so that the water column will rise up like this. This is H2, this is H1. This will go down like this. Okay. So here volume of CO2, sorry, volume of oxygen absorbed here is equals to H2 minus H1 into A. Rate of respiration is equals to, we assume that rate of respiration is equals to rate of absorption of oxygen or rate of carbon dioxide release. Here from oxygen we can take. So the rate of absorption of oxygen here, H2 minus H1 into A divided by time taken. Right? So this is the rate. Another. It's not asking the question, I'm just explaining. And the second part of this practical, you'll be removing the KOH tube. And then what you will be doing is, that means it's absorbing oxygen, releasing little amount of carbon dioxide. So this is not rising up like this. This will be finishing like this point. Okay. Right. So here, we oxygen minus V CO2 is equals to H3 minus H1 into A. Alright? So VO2 we already know. H2 minus H1 into A minus VCO2 is equals to H3 minus H1 into A. Another. So what we need is VCO2. You will get finally once cancelling all the cancelling off is done. H2 minus H3 into A is VCO2. So you can get the rate of respiration using this value as well. What we are going to finally find in this question is respiratory quotient RQ is equals to VCO2 divided by VO2. So here VCO2 is H2 minus H3 into A divided by H1, so H2 minus H1 into A. A will be cancelled off, so you will get this answer. So this is equals to 1 since the respiratory quotient, the respiratory substrate here is carbohydrate. For carbohydrates, respiratory quotient value is 1. That means in this one, you will be getting equal volume of carbon dioxide and equal volume of oxygen. If it is proteins, RQ value is 0.8. If it is lipid, RQ value is 0.7. Please write this down and write the whole procedure and keep it. Go home and read the practical again. If you don't understand, ask again, okay? Finish writing? Right, let's go to the next one. What are the two observations that Charles Darwin made that enabled him to put forward the theory of natural selection? Number one, variation. Number two, overproduction. Give two characteristics that are favorable for survival and reproduction. Escaping from predators, or else defense, tolerating physical conditions. Number three, obtaining food. Number four, resistance against diseases. Number five, fertilizing probability. Or number of offspring produced. Hi. Name three structures that amphibians utilize for gaseous exchange. So they will use lungs. Main thing is lungs, okay? Number two, they will be using their moist skin, or else they will be using their buccal cavity or oral cavity lining. Tadpoles like ones are having gills too. Correct? Asking the differences between osteichthyes and chondrichthyes. So their skeleton is predominantly of cartilages. Here the skin is mainly of bones. That's the first difference to write. Then here the caudal fin is heterocircle. Here the caudal fin is homocircle. Better if you can write as sentences. Then chondrichthyes are having placoid scales. Here ctenoid or cycloid scales are there. Gills are not covered by an operculum. Here, gills are covered by operculum. Correct? Fan එක off කරන්න ඕනද? Right, next one. There's a dichotomous key given here. Are they erasing this one? Are you all done writing with this? Online students, are you done? I'm erasing. So we have now limited time is available, and tell if you have any friends about the class, about the paper class that we'll be doing here. Tell them to join sooner, otherwise we don't have time, you know, like how much of big amount of work that we have already done. Those who have joined early, you will understand what's the importance of that. Then homosporous. Who are the homosporous ones here? Prognathum and Nephrolipis are homosporous ones here. Since we have two pute, another vidura, we can't write the names here. When we have more than two, we are not going to write the name here. So the first category, homosporous, there are two types of ones. So we will be sending them into the second category. Put in number 2, denote in that you will be sending them to the second category to divide. So heterosporous. Heterosporous, once others are heterosporous, so we will be sending them to the third category, right? Look at this one now. Number 2, underground rhizome present in Nephrolipis only. So we can write the name since there is only one. Our scientific names always should be underlined, even if it is this kind of a question. You have to underline. And then underground rhizome is absent. In Prognathum. Alright. So this second one, number 3. Pollen grains and sperms are produced from the same. From the ones you send to the third category, only dividing from here onwards. Pollen grains, sperms produced. Remember very well, if you have got some place wrong over here, all other answers also wrong here. You can't get marks. The rest of the answers, if it is correct, but still you are not getting marks, okay? If one place is wrong, means whole dichotomous key is wrong. Not produced. So among the remaining ones, pollen grains are produced in netum, shoe flower, and cycas now. So we can't divide them. So we will be sending them to the fourth category. But pollen grains are not produced only in Selaginella from the remaining ones. So we can write Selaginella name here. Then number 4, vessel elements present in the xylem or absent. Vessel elements present in the xylem, they are having netum and show flowers. So we'll be sending them to the fifth one. Show vessel elements are absent in cycas. So we'll be writing cycles here. And fifth category, we have shoe flower and netum. Seeds found enclosed within a fruit. Shoe flower. It's a common name, you don't need to underline. Seeds found not enclosed within the fruit. Neat. Alright. I'm giving an example for you to try alone. You need to make a dichotomous key to this one. And remember very well, mostly you'll be considering the external appearance or the features that you can see from outside, the morphological features for a dichotomous key. Use these organisms and make a dichotomous key. This is one you need to make. Number two also I'll give. Wait, that side is not visible. ఎవరిది కుత్తే విదురు ఫినిష్ Tharanga. See the board now? Sorry, now only I saw the message. Can everyone see the board properly? Are you all done? Tilki, are you finished? Lausilu? And Aravendi? And okay, right. So we'll discuss that one. First one first. You can use any of the features of them. I'm taking one. It doesn't. Your one doesn't have to be my one. Check if it is correct. Correct. I'm firstly taking the throat is given here. First category I'm taking unicellular. Sorry for the interruption. Those who couldn't hear me, I just did the dichotomous key that I have given. Sorry, my mic was not working. Now okay, now you can hear me now. Right, write this down if you have not done it properly. This doesn't have to be the same as what I have done here. It is, you can use any of the categories, but don't use the things such as these chlorophyll and those pigments are present, absent, like things you can't use. Other things, any of the external morphological appearance, you can take it and do the dichotomous key. Okay, the pute vidura. Alright, let's do one for the next one as well. For the others, is it okay to erase this one? I'm erasing. I'm going for the next example also. Butterfly, donkey, snail, sea star, jellyfish, crow, and tapeworm. Let's see what we can do here. Here also range of things you can take in order to divide. I'm just taking few things and divide it. Going. I'll take first presence of wings and absence of wings. Number one, presence of wings. Absence of wings. Right? Presence of wings, butterfly and crow are having wings here. So we'll be sending them to the second category as we can't divide. So number two in the second category, butterfly and crow is there. So we can say that endoskeleton is present, endoskeleton is absent. Endoskeleton is present in crow. Endoskeleton absent in butterfly. Select number three, we'll be sending them into the third category, the remaining ones. So we can say radial symmetry is present. Radial symmetry is absent. So we will be taking two people here, jellyfish and the sea star, because pentaradial is also type of radial symmetry. So we can't divide them here. So we'll be sending them into the fourth category. So in the fourth category, we have two people. So among that, endoskeleton is, water vascular system is present, absent. Present in sea star. Water vascular system is absent in jellyfish. So those are done now. So we'll be sending them to the fifth category. These guys, fifth category. So you have now remaining donkey, snail. Donkey is there, snail is there. Tapeworm is remaining. No, all others done. No? So having legs for locomotion. Not having legs for locomotion. Having legs for locomotion here, donkey. The other two are not having, so we'll be sending them to the sixth category. In the sixth category, we have now snail and the tapeworm. Among that, dorsoventrally flattened body. having a dorsoventrally flattened body. Who is not having dorsoventrally flattened body? Who is having tapeworm and not having dorsoventrally flattened body? It's snail, right? If you want, you can write this down quickly. Otherwise, you make one of your own, right? You don't have to depend on my one. Okay, Putti, let's go to the rest of the questions in the paper. Asking what is a meristem? Undifferentiated tissue groups in a plant body. We can say that like this way also. It's a type of undifferentiated tissue in plant body. The next one, state a functional characteristic feature of meristematic tissue, ability to multiply. Number 3, name two types of apical meristems, shoot apical meristem and root apical meristem. Next one, state two structural differences between the tissues mentioned in 3 above. Structural difference. Shoot apical meristem can be found at the tip of the shoot. Root apical meristem can be found at the tip of the root. Next one, shoot apical meristem is protected by leaf primordia and the root apical meristem is protected by the root cap. Right? Draw a label diagram of a cross section of primary structure of a monocot root. So here is how we need to draw. This is the outermost layer. Epidermis. Epidermis is having root hair cells since this is a root. So cortex is here. Inner to the cortex, endodermis is there. Endodermis is 1 cell layer thickness. And inner to that, pericycle is there. And the xylem and phloem, alternatively like this, they can be seen. So root hair, epidermis, cortex, endodermis, pericycle, xylem, phloem, pith. Alright, have you done properly? All done, others? Next question is asking, what is the reproductive structure of flowering plants? Flower. Name 4 whorls of leaves. It's composed of petals, sepals, stamens, and carpels. Petals, sepals, stamens, and carpels. What do the following structures of cycas transform into after fertilization? Female gametophyte will be producing 2N endosperm. Ovule is, that is wrong, neither not 2N endosperm is produced by this one. This polar nuclei will be becoming 2N endosperm. Something is wrong there in this question. Ovule is becoming the seed, that is true. What is known as, cut the first A1 there in that one is wrong. What is known as photoperiod of a plant? The interval of 24-hour period in which plant gets exposed to light. Interval of 24-hour period in which plant gets exposed to light. Alright. Next one. The following questions are regarding the experimental procedure of determining the solute concentration of rohioepidermal cells. What is a fully turgid cell? Fully turgid means its cell is placed inside pure water, so that cell has now come to an equilibrium with pure water. So that cell's psi value has now become 0. So psi of cell is 0. Therefore, psi s of cell is equals to psi p of the cell. You can say that cell has maximum psi p. What is the reason for using lower epidermal peels for this experiment? because of the anthocyanin pigment present in the vacuole, the turgid and facet cells can be observed clearly, or else you can say that plasmolized cells can be observed clearly because of the anthocyanin pigment present in the vacuole or the cell sap. Next one, how long you have to keep? 20 minutes. A graph x-axis and y-axis is asked here. X-axis is concentration or the molarity of the sucrose solution. Better to write molarity. Y-axis percentage plasmolysis. Alright, percentage plasmolysis. Next one, name two types of fibers present in areolar tissue. Collagen fibers, elastin fibers, and reticular fibers. State a function of each fiber named in part 1. Collagen fibers provide strength and flexibility. Elastin fibers makes tissues elastic. Reticular fibers join connective tissues of adjacent tissues. C. State two unique features of blood compared to the other connective tissues. Extracellular matrix is liquid. Matrix is not secreted by its cells. Fibers are formed only when blood clotting occurs. Among that, the last two is the better answers. Matrix is not secreted by cells and fibers are formed only during blood clotting. Show the stages of holocyte nutrition using a flow chart. Number 1, ingestion. Then digestion. Absorption. Assimilation. Elimination. Next one. State one function for each of the following components of saliva. First one, water. You can say liquidify food. Else you can write provide watery medium for chemical digestion. Next one, aids in taste reception. Mucus make it easier to swallow. Or else clean the mouth. Else you can write protect the lining of the mouth from abrasion. Name an antimicrobial substance that is present in saliva, immunoglobulin or lysozymes. State three structures that can be found at the corner of a liver lobule, a branch of a hepatic artery, branch of hepatic portal vein, intralobular bile duct. Three things again I'm repeating. Branch of hepatic artery, branch of hepatic portal vein, intralobular bile duct. Alright. Explain how absorption of the end products of the fat digestion into blood occurs in the small intestine. We can say end results of the fat digestion of fatty acids, glycerol and monoglycerides. Fatty acids and monoglycerides are absorbed into the cell through microvilli. Within the cells, triglycerides are formed. They are incorporated into water-soluble globules called chylomicrons. Chylomicrons are transported into the lacteal and into the blood vessels through lymph. Do I need to repeat again that one? All right, number 1. Okay, I'll write it here. Write down the correct answer there. I'm writing the second question for the day. You had to write it after the class and submit to me. So write down the second question also. Describe the linear electron flow that takes place in the chloroplast during light reactions of photosynthesis. We briefly discuss the reasons for high efficiency of photosynthesis in C4 plants. Write down the question. Did you write? Hi. Now you have to do me a favor. Dilki has done this activity before. Others also, you might have several different sections in the lessons. No, Vidura, you also go home and send me a list of things that you feel difficult, so that time to time when I'm going through the revision, I'll take them. Okay. And Onit and Losilu and Aravindi, if please make a list of things that you feel difficult. Please send it to me in the WhatsApp. So I'll consider them, and time to time I'll be doing certain things. I think Dilki, some of your difficult list also I have covered so far. Neither. Did I? Okay, so then I'm finishing the class for the moment. Others wait in the class and write the question and give it to me. Losilu, Onef, Dilki, Aravindi, send me the answers after the class. Okay, only for Dilki has given chance to submit it later. Others have to submit it sooner. Okay, right. So everyone, good evening and have a nice day, everyone. Good afternoon, all.